Have you ever had one of those days where you feel like your brain is just totally stuck in its ways?
Oh, yeah, absolutely.
Like maybe you are sitting there right now trying to figure out why you cannot seem to break that one stubborn bad habit.
Or trying to learn something new and it isn't clicking.
Right, like a new language or some new software at work, and you feel like the information is just bouncing off a brick wall in your head.
It's frustrating.
It is. Or even more frustrating, maybe you are someone who finds yourself lying awake at 2 a.m. overthinking the exact same scenarios night after night.
The classic 2 a.m. doom loop.
Exactly, and you're wondering why you cannot just flip a switch and turn it off.
It's an incredibly common feeling, right? That sense of being trapped by your own neural wiring.
Yeah,
We often view our brains as these rigid computers that we are just stuck with once we hit adulthood. Like the hardware is locked in.
but it's not.
No, it's not. The reality of what is happening inside your head is so much more dynamic and honestly so much more hopeful than that.
Which is exactly why we are here today. We want to bring you, the listener, right into the middle of a conversation that is going to completely dismantle that feeling of being stuck.
Consider yourself the third person sitting at the table with us.
Main B is conversation cup heart hoon. That's the vibe we want. So welcome to another deep dive in our brain series.
It's going to be a good one.
Today, we are opening up a massive stack of cutting-edge neuroscience papers, detailed medical texts, and recent clinical reviews. And they all point to one truly miraculous, mind-bending reality about the human brain:
Neuroplasticity.
neuroplasticity.
It is no exaggeration to say that this is the absolute foundational concept of modern neuroscience.
Yeah.
We are going to explore the literal biological machinery behind how you learn, why certain memories stay with you for a lifetime.
While others fade in a day.
Right. And how the things you do constantly, like how many hours you sleep, whether you took a walk this morning, or even the chemical makeup of the coffee you might be drinking right now.
I am definitely drinking coffee right now.
Well, it is actively, physically remodeling your neural circuitry at this very second.
And I want to set some ground rules before we get into the weeds of the science here. This is not going to be some dry academic lecture where we just rattle off terms from a textbook.
Definitely not.
We are taking a deep dive into the practical reality of your own mind. We're going to look at the dark side of this too.
Because there is a dark side.
What happens when the brain's adaptability actually works against us, trapping us in cycles of stress, trauma, or addiction?
Or even just procrastination.
Oh, we are definitely getting into procrastination. But we're also going to look at the escape routes. We're going to unpack how the latest science shows we can actively rewire our brains for recovery.
Which is where the real power of this knowledge lies. Because once you understand the mechanism of how your brain changes itself, you start to realize how much agency you actually have over your own cognitive and emotional health.
So let's start at the beginning. I think to really appreciate how groundbreaking this all is, we have to look at what scientists used to believe.
The old dogma.
Yeah. For the longest time, the overwhelming consensus in the medical and scientific communities was that the adult brain was basically a fixed piece of hardware.
Right.
They called it the static brain dogma. What was the actual thinking behind that?
So the static brain dogma was a very deeply entrenched belief. It dominated the 19th and much of the 20th century.
Wow, that long.
Oh, yeah. The idea was that the brain was like a complex machine that got fully assembled during your childhood. By the time you reached your early 20s, the concrete had essentially dried.
So you're just done.
You were done. You were given a certain allotment of neurons. They were wired in a very specific, unchanging configuration, and that was the end of the story.
It sounds incredibly bleak.
It really is.
It basically implies that if you have a certain temperament or if you suffer some kind of brain injury, there is absolutely zero hope for change or recovery.
It was a very deterministic way of looking at human biology.
Yeah.
The prevailing thought was that if you lost brain cells, whether through aging, trauma, or disease, they were gone forever.
Just a slow, inevitable march toward cognitive decline.
Exactly. But what is truly fascinating is that the pushback against this idea started far earlier than we usually assume.
Really?
Yeah. We tend to think of neuroplasticity as a super modern 21st century buzzword, right?
Yeah, totally. It feels very new.
But the origins of challenging that rigid dogma actually date back to the late 1800s.
Wait, the 1800s? They barely had light bulbs, let alone functional MRI machines. how were they studying brain adaptability back then?
They weren't looking at the cellular level, of course. They couldn't. They were observing behavior.
Oh, okay.
The term plasticity in a psychological context was actually first coined by the pioneering psychologist William James.
Okay.
In his 1890 book, The Principles of Psychology, James was a brilliant observer of human nature.
So what did he notice?
He watched how people formed habits, how they learned new skills, and he reasoned that for our behaviors to be this flexible, the organic material of the brain itself had to be yielding to influence.
That makes a lot of logical sense.
Right. He wrote that the structure of the brain was, and this is a quote, weak enough to yield to an influence, but strong enough not to yield all at once.
That is a beautiful way to put it.
Isn't
It
it?
has to be malleable enough to learn, but stable enough so you don't forget who you are every time the wind blows.
Exactly.
But if William James was saying this in 1890, why did the static brain dogma persist for another century?
Because in science, observation isn't always enough to overturn an entrenched paradigm. You need a mechanism.
You have to show the receipts.
You need hard biological proof. And that is where things got really interesting in the early 1900s with the psychiatrist named Adolf Meyer.
Adolf Meyer.
Yeah, Meyer is a massive figure in the history of this field, though he doesn't always get the mainstream recognition he deserves. He championed a radically different holistic approach that he called psychobiology.
Psychobiology. That sounds like he was trying to merge two fields that absolutely did not want to be merged at the time.
That is precisely what he was doing. At that time, psychiatry and neurology were separated by a massive, rigid wall.
Right, the mind versus the brain.
Exactly. Neurologists dealt with the physical tissue of the brain, and psychiatrists dealt with the abstract realm of the mind. Meyer looked at that division and essentially said, this is nonsense.
Good for him.
He proposed that your mental and behavioral responses are directly physically linked to biological changes in your brain.
So he was arguing that your subjective experiences like the stress of your job, the grief of losing a loved one, or the joy of learning a new skill weren't just floating in some invisible mind cloud.
Right.
They were physically altering the meat of the brain.
Yes. He referred to the nervous system as the apparatus of biological plasticity.
Wow.
He argued that the nervous system wasn't a closed, static loop, but an integrated, open-ended living process that was in a constant state of negotiation with the environment.
How did the medical establishment react to that? I imagine if you walk into a room of 1920s neurologists and tell them that a patient's sad thoughts are physically reshaping their neural tissue, they probably wouldn't invite you back for dinner.
You are absolutely right. Many of his medical colleagues dismissed his ideas entirely.
Not surprising.
They found his theories overly convoluted, unorthodox, and bordering on the mystical because he lacked the experimental imaging to back it up.
He didn't have the tools.
Right. But Meyer remained steadfast, and as the decades rolled on, technology slowly caught up to his theories.
Right, we started getting better microscopes and imaging.
Yeah, in 1948, a Polish neuroscientist named Jerzy Konorski advanced the specific concept of neural plasticity, theorizing how neurons might actually change their connections.
Okay.
But it wasn't until the late 20th century, with the advent of advanced neuroimaging and cellular biology, that the dam finally broke.
And when it broke, we realized that the brain isn't just slightly flexible. It is constantly tearing itself down and rebuilding itself from the cradle to the grave.
Constantly.
And the research we are looking at categorizes this adaptability into two main buckets, right? Structural plasticity and functional plasticity.
Yes.
Break those down for us. What is the difference?
It comes down to the scale at which the change is happening. Let's look at structural plasticity first.
Okay.
This refers to the actual physical, architectural changes happening in the brain at the microscopic level. We are talking about the individual neurons themselves.
So the actual hardware is changing shape.
Exactly. When you learn something new, your neurons grow new appendages called dendritic spines to reach out and connect with other neurons.
Like branches on a tree.
Yes, it is called axonal sprouting. And it even goes as far as neurogenesis, which is the birth of entirely new functional neurons, particularly in an area of the brain called the hippocampus.
Which is the memory center.
Right, so structural plasticity is the literal building of new microscopic infrastructure.
Okay, so if structural plasticity is the microscopic building of roads and bridges, what is functional plasticity?
Functional plasticity operates on a macroscopic level. It is the brain's ability to actually move functions from one area of the brain to a completely different area.
Wait, move functions?
Yes. If a specific part of the brain is damaged, or if a particular sensory input is completely cut off, the brain can reorganize its entire network layout to compensate. It is a massive cortical remapping.
And there was a specific example of this functional remapping in the research that I had to read 3 times because I could not believe it was real.
I know exactly what you're talking about.
It involves human echolocation.
Ah, yes. This is arguably one of the most striking and beautiful illustrations of functional plasticity in all of neuroscience.
It really is. So for anyone listening who hasn't heard of this, there are individuals who are completely blind, but who have taught themselves to navigate their environment by making sharp clicking sounds with their tongues.
Yes, tongue clicks.
They listen to the way the sound waves bounce off objects in their environment and return to their ears. And the resolution they get from these echoes is staggering.
It's unbelievable.
They can ride bicycles, play basketball, navigate crowded city streets. They are essentially using sonar, just like a bat or a dolphin.
The behavioral adaptation alone is incredible, but what the neuroimaging reveals is where the real paradigm shift happens.
Right, because they finally scanned their brains.
Yeah. In 2010 and 2011, researchers decided to put some of these expert echolocators into functional MRI machines to see what was actually happening in their brains while they processed these click echoes.
And the logical assumption would be that their auditory cortex, the part of the brain that handles hearing, would be working in overdrive.
Right.
Because they are relying entirely on sound.
That was the assumption, but that is not what the fMRI scans showed.
This is the crazy part.
When these individuals listened to the echoes bouncing off virtual objects, the activity didn't just spike in the auditory regions. The massive surge of processing power was actually occurring in their visual cortex.
The visual cortex, the part of the brain at the very back of the head that is entirely dedicated to processing light and sight.
Yes. The visual cortex in these individuals had been entirely deprived of optical input. But the brain is an incredibly efficient organ.
It doesn't want to waste space.
It does not let prime processing real estate sit dormant. Because the visual cortex is essentially a supercomputer designed to process complex spatial relationships and map out three-dimensional environments, the brain completely remapped its internal routing.
It hijacked it.
It hijacked the visual cortex and repurposed it to analyze the spatial data coming in through the ears.
So they are literally seeing the world, but the data feed is acoustic instead of optical.
Precisely. The click echoes are providing a form of vision through sound.
That is just wild.
It completely shatters the old static brain dogma. The brain isn't rigidly compartmentalized with fixed borders. It is a maneuvering, highly dynamic mass that will completely rewrite its own operating system to help you survive and adapt to your environment.
It is a massive realization. But it also opens up a really dense biological mystery for me.
What's that?
If the brain is capable of changing its entire macroscopic layout, how does it actually do the day-to-day work? Like, how does it store a single tiny memory at the microscopic level?
Right.
If I learn to juggle today, how does my brain physically hold on to that new skill so I don't have to relearn it from scratch tomorrow?
To understand that, we have to zoom all the way back down to the microscopic level. We have to look at the foundational rule of neural networking, which was proposed by a psychologist named Donald Hebb in 1949.
Okay.
It is often summarized with a very catchy phrase, neurons that fire together wire together.
Neurons that fire together wire together.
This is known as Hebbian plasticity.
Let's ground this in a real-world example so you listening can really see it working. Let's say I am trying to learn a chord on a guitar, and I have never played it before.
Okay, perfect example. When you first try to place your fingers on the frets and strum the strings, it feels incredibly awkward, right?
Yeah, my fingers don't want to bend that way.
Your brain is trying to coordinate the visual input of seeing the strings, the tactile input of feeling the metal wire, the motor commands to move your fingers into that weird claw shape, and the auditory input of the sound it makes.
And usually the first sound it makes is terrible. Just a dull thud.
Right, because the neural networks coordinating all those different areas are very weak. They have never had to communicate in that specific pattern before.
They're strangers.
But as you practice, you force the neurons controlling your fingers and the neurons processing the sound to fire their electrical signals at the exact same time. You are repeatedly stimulating them simultaneously.
Okay.
And because they are firing together, the biological connection between them begins to physically strengthen.
So they recognize that they're part of the same team now.
Exactly. And the scientific term for this strengthening process is long-term potentiation, or LTP.
LTP, got it.
LTP is the persistent increase in synaptic strength following high-frequency stimulation. The synapse is the tiny gap between the neurons where they pass chemical messages to each other.
So the message gets across easier.
As you keep practicing that guitar chord, LTP is making that specific synapse more and more efficient. The signal travels faster and easier. Eventually, you can play the chord without even looking at your hand. The memory has been structurally encoded.
That makes perfect sense for learning. We want LTP to build strong connections. But the research also emphasizes the opposite process, something called long-term depression, or LTD.
Yes.
And I want to be clear for anyone listening, when we say depression here, we are not talking about the mood disorder, right?
Correct. In neurobiology, long-term depression refers to an activity-dependent reduction in the strength of neuronal synapses. It is the active weakening of connections.
But why would our brains ever want to actively weaken a connection? Wouldn't the goal of a healthy brain be to have everything perfectly strongly connected, like a super network?
It seems counterintuitive, but if your brain only ever experienced LTP, it would be catastrophic.
Really?
Think about it. If every single connection were continuously strengthened every time you experienced anything, your brain's networks would quickly become completely saturated.
Oh, just too much information.
You would be overwhelmed by metabolic noise. You wouldn't be able to distinguish important information from trivial background details. You wouldn't be able to break a bad habit because the old network would never weaken.
Oh, wow. I never thought of it that way. So if I parked my car in a different spot at the grocery store today, but my brain couldn't weaken the memory of where I parked it yesterday, I would be wandering the parking lot forever.
Precisely. You need to forget the old spot. LTD allows the brain to prune away irrelevant or outdated information.
So forgetting is a feature, not a bug.
It is the biological mechanism of forgetting, which is just as vital as remembering. Think of the brain as a sculptor working with a block of stone.
Okay.
LTP is the process of adding clay to build up a structure, to emphasize certain features. But LTD is the chisel, chipping away the excess stone to reveal the actual masterpiece underneath.
That is a phenomenal analogy.
Without LTD, you don't have a statue, you just have a massive, formless block.
You need both the adding and the taking away to create meaning. This actually explains so much about why our brains overthink, doesn't it?
How do you mean?
Well, if someone is lying in bed overthinking a conversation they had 5 years ago, they are stuck in a loop. It's almost like their brain is failing to apply the chisel. The LTD isn't kicking in to weaken that useless, anxious memory.
That is a very astute connection. When the pruning mechanisms fail, or when a network is overly strengthened by emotional arousal, you get those rumination loops.
But what actually governs that? How does the brain know when to use the clay and when to use the chisel?
That delicate balance is governed by two overarching systems, metaplasticity and homeostatic plasticity.
Okay.
Metaplasticity is a bit of a mind bender. It is essentially the plasticity of plasticity.
Okay, you're going to have to slow down and unpack that one.
Think of it like a thermostat for your ability to learn. Metaplasticity regulates how easily a specific synapse can undergo LTP or LTD based on its prior history.
So it remembers how much it has already changed.
Yes. If a neuron has been highly active for a long time, metaplasticity might adjust the threshold, making it harder to induce more LTP, preventing it from maxing out. It shifts the rules of the game based on what has already happened. Homeostatic plasticity, on the other hand, is about the whole network. It ensures that the overall electrical excitability of a neural circuit remains within a safe functional range.
So it doesn't short circuit.
Exactly, preventing the network from becoming dangerously hyperactive, like in a seizure, or completely silent.
So we have this incredible self-regulating system of building and pruning. But I want to zoom in even closer.
Let's do it.
We've talked about neurons passing messages across the synapse, the gap between them. How does a synapse actually physically get stronger? The research papers describe this using a phrase that sounds incredibly intimidating: the glutamatergic calculus.
It sounds like an advanced math class you want to drop on the first day, I know.
It really does.
But it is actually a beautifully elegant piece of biochemistry. It primarily revolves around a chemical called glutamate. Glutamate is the brain's most abundant excitatory neurotransmitter. It is the chemical spark that tells the next neuron in line to fire.
Okay, so neuron A wants to send a message to neuron B. It spits glutamate into the gap between them.
Exactly. And that glutamate floats across the gap and binds to specific receptors on the surface of neuron B.
Like catching a baseball.
You can think of these receptors as specialized locks, and glutamate is the key. In this process, there are two main types of locks we need to care about: AMPA receptors and NMDA receptors.
AMPA and NMDA.
And these two receptors have very different jobs.
If I'm reading this right, the AMPA receptor is like a standard revolving door. Glutamate hits it, it opens, the signal goes through.
Right.
But the NMDA receptor is much more stubborn. It has a literal physical plug stuck in it.
Yes, that is a fantastic way to visualize it. Under normal resting conditions, the pore of the NMDA receptor is completely blocked by a single magnesium ion.
Magnesium, like the mineral in my daily vitamin.
The very same. Because of the electrical charge of the resting neuron, this positively charged magnesium ion is drawn into the channel of the NMDA receptor, effectively plugging it up like a cork in a wine bottle.
So it's physically stuck in the doorway.
Even if glutamate is binding to the outside of the receptor, the door cannot open because the magnesium plug is in the way.
So how does the brain ever form a memory if the crucial door is glued shut by magnesium? What pops the cork?
This is where the AMPA receptors do their heavy lifting. When neuron A fires a little bit of glutamate, it binds to the AMPA receptors, opening them up and allowing a small amount of positive charge into neuron B. This causes a slight depolarization, a shift in the electrical voltage of the cell membrane. But if neuron A fires a lot of glutamate, because you are intensely focusing on that guitar chord, repeating it over and over,
The AMPA receptors are working overtime.
they let a massive wave of positive charge into neuron B.
And because magnesium is positively charged, and like charges repel each other.
Exactly. The sudden rush of positive charge inside the cell violently repels the positively charged magnesium ion. It literally pops the plug out of the NMDA receptor.
That is amazing. The sheer electrical force of your focus physically ejects the blockage.
It's mechanical and electrical all at once.
What happens once the door's open?
The moment that pore is open, calcium floods into the cell. And calcium is the golden ticket.
What does the calcium do?
It acts as a secondary messenger, triggering a massive cascade of biochemical reactions inside the neuron. These enzymes get to work immediately. They grab extra AMPA receptors that were hiding inside the cell and shove them up to the surface.
So they build more doors.
They build more doors. They also begin to physically alter the scaffolding of the neuron, enlarging the dendritic spine so it can catch more signals in the future. The connection is now structurally, physically stronger. You have just experienced long-term potentiation.
Okay, my mind is completely blown by the mechanics of that, but the research points out a catch.
There's always a catch.
All of that amazing calcium rushing in and pushing new receptors to the surface, that only creates a short-term memory, right? If the process stops there, the memory will vanish in a matter of hours.
That is the catch. The initial phase of LTP only uses proteins and materials that are already hanging around near the synapse. It is a quick and dirty reinforcement.
Like a temporary patch.
Exactly. But to stabilize those changes, to make sure you still remember that guitar chord next week, or to remember your wedding day decades from now, the neuron has to do something much more dramatic.
What does it have to do?
It has to synthesize entirely new proteins. And to do that, it has to send a signal all the way from the outer edge of the synapse, deep into the core of the cell, to the nucleus, to alter your actual gene expression.
And this introduces a whole new cast of microscopic characters. The sources call them immediate early genes, or IEGs. Specifically, they highlight one called ARC.
ARC, yes.
What makes ARC so crucial to our memory?
ARC stands for activity-regulated cytoskeleton-associated protein, and its role is absolutely vital for memory consolidation. Studies have shown that if you block the production of the ARC protein in a subject's brain, their neurons can still undergo that initial phase of LTP.
So they can pop the magnesium plug.
Right, they pop the plug, the calcium rushes in, they form the memory in the short term.
But what happens over time without ARC?
Without ARC, they cannot maintain it. The newly expanded scaffolding of the synapse is highly unstable. ARC acts as the molecular cement.
Oh, I see.
It binds to the structural proteins and locks them in place. Without that cement, the newly built structure quickly collapses, and the memory rapidly fades into nothing.
You could practice that guitar chord for hours, but without ARC stabilizing the synapse, you would wake up the next morning with absolutely zero retention.
It would be like you never played it at all.
So ARC is the cement.
Yeah.
But it gets even deeper than that. The deepest level of this rewiring goes down to the literal DNA inside our cells through a process called epigenetics.
This is a huge field right now.
I think a lot of people hear DNA and they think of an unchanging blueprint they inherited from their parents. How do our daily experiences rewrite our DNA?
This is truly one of the most profound frontiers in modern neuroscience. How do fleeting, intangible experiences, a conversation, a moment of fear, an hour of studying, become permanently etched into biological tissue?
How?
The answer is epigenetics. Epigenetics doesn't change the underlying genetic code. The letters of your DNA remain the same, but it changes how those genes are read and expressed.
Okay, so it changes the instructions.
It involves mechanisms like DNA methylation and histone modifications.
Let's break those down. What is DNA methylation doing during a memory?
DNA methylation involves attaching a tiny chemical tag-a-methyl group directly onto the DNA strand. Think of it like putting a piece of dark tape over a specific sentence in an instruction manual.
So you can't read that sentence anymore.
Exactly. When a gene is heavily methylated, it is typically silenced. The cell cannot read it. What researchers have discovered is that when you form a strong, long-term memory, there is a dynamic, highly targeted shift in these methylation patterns in your brain.
Can you give us an example from the research? How fast does this tape get applied?
The sources cite studies on contextual fear conditioning. That's teaching a subject to associate a specific environment with a mild stressor.
Like a shock?
Right. When the memory of that fear is formed, researchers observe a rapid and long-lasting increase in DNA methylation in the prefrontal cortex.
How long lasting?
This chemical tagging persists for over 30 days. The memory of the fear is quite literally locking down the expression of specific genes to ensure the brain remembers the danger.
And the other mechanism, histone modification, works a little differently.
Yes, your DNA is incredibly long. To fit inside the microscopic nucleus of a cell, it has to be tightly wound around spool-like proteins called histones.
Like thread on a spool.
Exactly. If the DNA is wound too tightly, the cellular machinery cannot get in to read the genes. Modifying these histones, specifically through a process called acetylation, essentially loosens the spool. It relaxes the structure, exposing the DNA.
So methylation is taping over the instructions, and histone acetylation is opening the book wide so the cell can read it.
Precisely. And when the spool is loosened, memory-related genes, like the ones that produce BDNF, a crucial growth factor we will discuss later, can be actively read and transcribed.
Wow.
So, epigenetics provides the molecular ledger. It is the ultimate accounting system that maintains stable, long-lasting changes in your brain over time.
It is a breathtakingly beautiful system when it is working in our favor. When you are learning to paint, or memorizing a poem, or remembering a first date,
It's elegant.
you focus your attention. The AMPA receptors flood the cell, the magnesium plug pops, the calcium rushes in, the ARC protein pours the cement, and the epigenetic tags lock the memory into your very DNA.
That's the ideal scenario.
But as we promised at the beginning, this is not a fairy tale. Neuroplasticity is value neutral.
Completely neutral.
It doesn't care if the environment it is adapting to is healthy or toxic. It just adapts. And that brings us to the dark side of this science.
Yeah, this is where things get heavy.
What happens when the environment forces the brain to adapt to chronic stress or trauma?
This is where we see the double-edged sword of neuroplasticity. The brain is an incredibly sensitive instrument. When an individual is subjected to chronic stress or experiences adverse childhood events, which the literature refers to as early stress, the brain's incredible capacity to adapt actually becomes maladaptive.
It backfires.
It begins to wire itself for pure survival at the expense of higher cognitive function and emotional stability.
And the main culprit driving this negative remodeling is a system we all have, the HPA axis.
Yes, the hypothalamic pituitary adrenal axis. This is your body's primary stress response system.
How does it work?
When you perceive a threat, this axis activates, setting off a cascade of hormonal signals that culminates in the release of glucocorticoids from your adrenal glands. The most famous of these is cortisol.
Now, I feel like cortisol gets a really bad reputation in the wellness world.
It really does.
But cortisol isn't inherently evil, right? We literally need a spike of it every morning just to wake up, or to hit the brakes if a car suddenly swerves into our lane.
Absolutely. Acute short-term stress responses are highly adaptive. They flood your body with energy, sharpen your focus, and prepare you to fight or flee. Without cortisol, we wouldn't survive.
So when does it become a problem?
The pathology begins when the stress doesn't stop. When the HPA axis is constantly engaged day after day, month after month, those high levels of cortisol become profoundly neurotoxic.
Neurotoxic, they poison the brain.
Yes, and they do not affect the entire brain equally. This is what we often refer to as the tale of two brain regions, the hippocampus and the amygdala.
Let's start with the hippocampus. We know it is the primary center for learning and memory. How does chronic cortisol exposure physically damage the memory center?
The hippocampus is exquisitely vulnerable to stress because it happens to have a very high concentration of receptors for cortisol.
So it absorbs a lot of it.
It absorbs the brunt of the hormonal flood. When exposed to high, sustained levels of cortisol, the neurons in the hippocampus undergo a terrifying structural change. Those beautiful branching dendritic spines that we just talked about building during learning, they actually begin to retract and wither.
So the physical connections you have built are literally melting away under the stress hormone.
Yes, the infrastructure crumbles. Furthermore, chronic stress actively suppresses neurogenesis in a specific part of the hippocampus called the dentate gyrus.
So not only are you losing the connections you have, but your brain's ability to generate new neurons to replace them is severely impaired.
The hippocampus physically shrinks in volume.
That is devastating. But the amygdala, which is the brain's fear and emotional processing center, is having the exact opposite reaction to the stress, isn't it?
It is. While the hippocampus is withering, the amygdala is thriving on the cortisol.
Like feeding a monster.
Under chronic stress, neurons in the basolateral amygdala actually exhibit enhanced dendritic arborization.
Which means?
They grow thicker, more complex branches, they form more connections. The amygdala becomes hyperactive and highly sensitized to any potential threat in the environment.
So this actually brings up a really interesting evolutionary point. This is why humans fear deep water, right? Or the dark.
That's a great connection, yes.
The amygdala is wired to keep us alive. So if you are a prehistoric human and you almost drown, the amygdala gets flooded with stress hormones and builds massive, thick neural connections to ensure you never go near deep water again.
Exactly, it's an evolutionary survival mechanism.
The way I always try to visualize this modern chronic stress is by imagining the brain as a massive, intricate city. Let's say the amygdala is the military command center, the panic room.
I like this.
The hippocampus is the grand, sprawling public library where all the history and context and knowledge is stored.
Right.
When the city is under constant, relentless siege from chronic stress, the male panics. They divert all the city's resources, all the funding, directly to the military center. The amygdala builds thicker concrete walls, installs thousands of hypersensitive alarms, and puts everyone on edge.
Preparing for war.
Exactly. But to pay for that, they completely defund the library. The roof of the hippocampus caves in, the books rot, the librarians are fired, and no new books are ever purchased.
That is painfully accurate.
The city becomes brilliantly, terrifyingly efficient at detecting threats, but it completely loses its history, its context, and its ability to learn anything new or see nuance.
That analogy captures the physiological reality perfectly. The brain is prioritizing immediate survival over long-term cognitive health. This maladaptive plasticity is the structural foundation of many severe mental health disorders.
And the sources specifically highlight borderline personality disorder, or BPD, as a profound manifestation of this.
BPD is incredibly complex.
How does this city under siege dynamic play out in the brains of individuals suffering from it?
BPD is characterized by intense emotional dysregulation, compulsive behaviors, and a very high vulnerability to stress. When researchers use MRI technology to look at the brains of patients with BPD, the structural abnormalities are stark.
What do they see?
They have documented up to a 24% loss in hippocampal volume compared to healthy controls.
24%. A quarter of the memory and context center is just gone.
It is a massive reduction, which helps explain the difficulties with memory integration and emotional context. But what is truly surprising in the BPD data is the amygdala.
Okay, what about it?
They also report an amygdalar volume reduction of up to 20%.
Wait, hold up. You just said that chronic stress makes the amygdala grow larger and hyperactive. Why is it shrinking in BPD patients?
It is a crucial distinction that highlights the long-term trajectory of severe trauma. In the initial phases of chronic stress, yes, the amygdala undergoes hypertrophy, it grows and becomes hyperactive. However, in cases of severe, unrelenting, long-term trauma, which is often a precursor to BPD, the constant overactivation takes a toll. The neurons in the amygdala are firing so intensely and so constantly that they suffer from excitotoxicity.
Cytotoxicity. So they basically burn themselves out from overuse.
Precisely. The calcium influx that we talked about being so crucial for learning, if it is too massive and unending, it becomes toxic. It triggers enzymes that actually destroy the cell from the inside out.
So they literally work themselves to death.
Eventually, the neurons die, leading to volume reduction. The entire regulatory system is burnt out. leaving the individual with profound emotional volatility and a desperate need to escape the internal pain.
And that desperate need for relief often leads to another form of maladaptive plasticity, which the sources cover in deep detail addiction.
Yes.
People turn to substances to quiet the hyperactive alarms. But what is happening neurologically? Why does the brain latch onto these substances so aggressively?
Addiction is perhaps the most tragic example of our learning mechanisms being completely hijacked. To understand it, we have to look at the mesolimbic system, which is the brain's primary reward pathway.
Okay.
The key players here are the ventral tegmental area, or VTA, and the nucleus accumbens, or NAC. And the currency of this system is dopamine.
We hear about dopamine all the time. Dopamine detoxes, dopamine hits from social media. The VTA is essentially the dopamine dispenser, right?
Basically.
Under normal circumstances, it gives us a little squirt of dopamine to reward us for doing things that keep the human race alive, like eating a good meal, exercising, or reproducing.
That is its evolutionary purpose. It highlights a behavior and says, that was good for survival. Remember how to do that again.
So this ties right into why we procrastinate, doesn't it?
It entirely does.
Like, if I have to write a hard report, that requires effort. But if I pick up my phone and scroll, my VTA gives me cheap, instant dopamine. My brain learns that the phone is a highly efficient reward, so it heavily weights that synaptic connection over the work.
That is a perfect everyday example of synaptic weighting. Your brain is optimizing for the quickest reward. But drugs of abuse fundamentally short-circuit this system on a much more massive scale.
How so?
Whether it is cocaine, opioids, or alcohol, these substances cause an unnatural, massive surge of dopamine in the nucleus accumbens, far beyond what any natural reward could produce. Some prevent the reuptake of dopamine, leaving it flooding the synapse.
So instead of a gentle pat on the back, the brain gets hit with a tsunami of reward signaling.
And the brain interprets that massive signal as the most important survival event that has ever happened.
Wow.
This intense surge artificially induces rampant long-term potentiation in the reward circuitry. It strongly, rigidly reinforces the drug-taking behavior, associating it with any environmental cue present at the time.
But the brain is a homeostatic organ. It does not like to be perpetually out of balance. If there is a massive flood of stimulation, it tries to defend itself.
Yes, it tries to scale the sensitivity back so the circuits don't literally fry.
How does it do that? The research focuses specifically on cocaine as an example, and it introduces a very unexpected mechanism involving the immune system.
This is one of the most fascinating discoveries in addiction research. We used to think addiction was purely a neurotransmitter issue.
Just a chemical imbalance.
Right. But the sources detail how repeated administration of cocaine actually activates microglial cells in a part of the brain called the striatum.
Microglia. What are those?
Microglia are the resident immune cells of the brain. They usually clean up debris and fight infections.
Wait, so the brain is treating the cocaine not just as a chemical, but as an actual physical infection that needs to be attacked.
In a sense, yes. When the microglia are activated by the presence of the drug, they produce and release a pro-inflammatory cytokine. Specifically, tumor necrosis factor alpha, or TNF-alpha.
TNF-alpha. Okay, what does an inflammatory immune chemical do to the reward circuitry?
It has a highly specific and devastating effect. TNF-alpha acts directly on the synapses in the nucleus accumbens. It drives the internalization of the AMPA receptors.
The AMPA receptors. The revolving doors we need to let the signal in to pop the magnesium plug.
Exactly. The immune response literally grabs those receptors from the surface of the neuron and pulls them back inside the cell, out of the synapse.
It takes the doors off their hinges and hides them.
Yes. This profoundly depresses the glutamatergic synaptic strength. It is deliberately weakening the connection to compensate for the massive drug high. It numbs the reward center.
So the brain is intentionally making itself deaf to the signal because the signal is too loud.
Exactly.
Which explains why people build a tolerance and need more and more of the drug just to feel normal, let alone get high. The doors have been removed.
Yes, that mechanism limits the initial behavioral sensitization. But here's the truly insidious part and why relapse is so biologically difficult to overcome.
Why is that?
Following a period of abstinence, say the person gets clean for a few months, their synapses are still in this weakened, altered state.
Yeah.
If they encounter a trigger, a place, a person, or even a small amount of the drug, those sensitized microglia reactivate immediately. They dump more TNF-alpha, further depressing the synaptic strength and triggering intense craving and a massive behavioral response.
So the neuroplastic architecture, the entire reward system, has been fundamentally, structurally altered by the inflammatory immune response.
It is terrifying. It strips away all the moral judgment around addiction and shows it for what it truly is, a profound physical alteration of the brain's hardware. The immune system literally tore down the receptors.
But if we know all of this, if we know that chronic stress, trauma, and addiction can physically damage and rewire the brain, it raises the most important question of this entire deep dive.
How do we fix it?
Exactly. How do we fix it? We
Yeah.
can't just talk away structural damage. How is modern medicine using neuroplasticity to rebuild the brain?
It is requiring a massive paradigm shift in psychiatry. For decades, the public and much of the medical field operated under the chemical imbalance theory.
The idea was simple, right? If you are depressed, you have low serotonin. Give a pill that boosts serotonin, problem solved.
But we are moving rapidly away from that oversimplified model. Depression, PTSD, and anxiety are increasingly viewed as failures of neural adaptability.
The brain gets stuck in a rut and loses the ability to pave new roads.
Yes.
The sources mentioned that severe depression is marked by a significant loss of dendritic spine density, specifically in the prefrontal cortex. That's the area behind your forehead responsible for complex thought, decision-making, and regulating those emotional alarms from the amygdala.
Correct.
So, if depression is a structural loss of connections, how do traditional treatments like SSRIs actually work? Because they do target serotonin.
They do target serotonin. SSRIs, or selective serotonin reuptake inhibitors like fluoxetine or sertraline, inhibit the reabsorption of serotonin,
Which means?
meaning there is immediately more serotonin floating in the synapse within hours of taking the first pill. But here's the paradox that baffled scientists for years. If it was just about correcting a serotonin deficit, the patient should feel better that same afternoon.
Because the chemical's there.
Because the chemical is there.
Well,
But anyone who has taken an SSRI knows that clinical relief of depressive symptoms usually takes weeks, sometimes 4 to 6 weeks, to kick in.
so what on earth is the brain doing during those 4 to 6 weeks?
The SSRIs are slowly, painstakingly restoring the brain's plastic potential. The increased serotonin is just the trigger. The downstream effect of that chronic serotonin elevation is what matters.
Okay, what is the downstream effect?
Over weeks of administration, SSRIs have been shown to significantly boost the levels of a crucial protein called brain-derived neurotrophic factor, or BDNF.
BDNF. This is the compound that scientists affectionately call the miracle grow of the brain.
And it earns that title. BDNF is a highly potent neurotrophin. It promotes the survival of existing neurons, prevents cell death, and critically, it encourages the growth and differentiation of new neurons and synapses.
Oh,
SSRIs actually stimulate neurogenesis, the birth of new brain cells in the adult hippocampus.
but building a new brain cell from scratch, letting it mature, and waiting for it to integrate into the existing neural networks takes time.
It takes weeks. That biological timeline aligns perfectly with the delayed onset of clinical relief.
So the pill isn't just a chemical band-aid. It is a construction foreman, initiating a weeks-long rebuilding of the shrunken hippocampus. That is amazing.
It is.
But it also highlights a major problem. What if someone is in a severe crisis? What if a patient doesn't have 6 weeks to wait for new neurons to grow?
That is the tragic limitation of traditional antidepressants.
Which leads us to what is arguably the most exciting breakthrough in modern psychiatry, which the sources cover extensively, ketamine.
Ketamine has completely shaken the foundations of psychiatric treatment for treatment-resistant depression.
How is it different?
It works on a completely different timeline and a completely different neurotransmitter system than SSRIs. While SSRIs target serotonin, ketamine targets the glutamatergic system.
The same glutamate system we talked about with the memory calculus.
The exact same one.
But the research says ketamine acts as an antagonist. It blocks the NMDA receptors. Wait, earlier we established that opening the NMDA receptor is exactly how we build memories and connections. Why would blocking the very door we need to open cure depression?
It sounds like a total paradox, I agree. But the genius of ketamine lies in exactly where it exerts its blocking effect.
Okay, where?
The brain is full of excitatory neurons pushing signals forward, but it also has inhibitory neurons, specifically GABAergic interneurons.
Okay, what do they do?
You can think of these interneurons as the brakes on a car. Their entire job is to suppress the release of glutamate and calm the system down. Ketamine preferentially targets and blocks the NMDA receptors on these specific inhibitory interneurons.
Oh, I see. It is blocking the brake.
Exactly. By blocking the brakes, ketamine takes the foot off the inhibitory pedal. This causes a sudden, rapid, and massive glutamate surge in the prefrontal cortex.
Prefrontal cortex gets flooded with glutamate. What does that surge do?
That massive wave of glutamate rushes in. and powerfully activates the AMPA receptors on the excitatory neurons. This massive AMPA activation triggers a specific molecular pathway inside the cell called the mTOR pathway.
The mTOR pathway, what is its job?
The mTOR pathway is a master regulator of protein synthesis. When ketamine kicks it into overdrive, it triggers explosive synaptogenesis. It rapidly rebuilds the dendritic spines.
How rapidly?
We are not talking about weeks here. Within hours of a ketamine infusion, researchers can observe the regeneration of synaptic connections in the prefrontal cortex that had been withered away by chronic stress.
That is legitimately science fiction. You are taking a brain that is physically damaged, shrunken by stress, and within a few hours, you are causing it to rapidly sprout a dense thicket of new connections.
It is a profound demonstration of how fast neuroplasticity can operate when the right molecular levers are pulled.
But pharmacology isn't the only way to induce these changes. The sources also discuss a fascinating structural barrier to plasticity called perineuronal nets, or PNNs.
Ah, yes, PNNs.
What are those? They sound like something Spider-Man leaves behind.
They do have a web-like quality. Perineuronal nets are specialized, dense structures made of extracellular matrix sugars and proteins. Think of them like a rigid physical mesh or scaffolding.
Okay.
During childhood, our brains are incredibly plastic. We absorb languages and skills effortlessly. This period is called a critical period. But as we transition into adulthood, the brain needs to stabilize all those networks it just built.
So they don't fall apart.
Right. So it secretes this dense mesh, the PNNs, which wrap tightly around certain neurons, particularly the inhibitory parvalbumin neurons.
So it is literally a physical cage that locks the neuron into place and says, okay, no more changing, this is who we are now.
Precisely. They act as molecular brakes, closing the critical window of high plasticity.
But in conditions like severe depression or PTSD, the brain is locked into a maladaptive negative pattern. The scaffolding is holding the trauma in place.
Exactly. So to heal, we might need to temporarily dissolve that scaffolding to reopen a window of juvenile-like plasticity.
And can we do that? Can we melt the cage?
Yes. Evidence suggests that part of the mechanism of various antidepressants, and even the effect of placing subjects in highly enriched, stimulating environments, is that they reduce the expression of these perineuronal nets.
Wow.
By softening that rigid scaffolding, the brain is returned to a more flexible state, allowing it to dismantle the maladaptive circuits and wire new, healthier ones.
And rewiring those healthier circuits is exactly what behavioral therapies do. The sources are very emphatic that we shouldn't view therapy as just talking about your feelings.
Not at all.
Modalities like cognitive behavioral therapy, or CBT, and mindfulness are actually driving physical, structural neuroplasticity.
Absolutely. CBT is, at its core, a highly structured, neuroplasticity-driven technique.
How so?
It focuses on identifying negative, automatic, catastrophic thought patterns and actively challenging them. For someone with depression, that their brain has highly efficient, thickly myelinated neural highways leading straight to despair and self-criticism.
Because they have traveled that road millions of times.
Right. Hebbian plasticity dictates that it is the path of least resistance.
Neurons that fire together, wire together, the depression highway's a 10-lane autobahn.
Exactly. CBT forces the patient to recognize when they are on that autobahn, hit the brakes, and deliberately take a different, smaller dirt road, a more logical, balanced thought.
That sounds exhausting.
At first, it is exhausting. The neural pathway for the balanced thought is weak, but every single time the patient actively reframes the negative thought, they are firing a new sequence of neurons.
Okay.
Through repeated practice, they induce LTP on the new pathway and LTD on the old one.
They are structurally dismantling the autobahn and paving a new highway.
Yes. Functional MRI studies show this beautifully. Successful psychotherapy physically restores the metabolic balance between different regions of the brain.
It changes the brain scans.
It does. It quiets down the ventromedial prefrontal cortex, which is often hyperactive and ruminative in depression, and it increases activity in the dorsolateral prefrontal cortex, which is responsible for executive control and emotional regulation.
And mindfulness practices do the exact same thing, right? Strengthening the insula and prefrontal networks to observe emotions without being hijacked by them.
Exactly the same principles.
And for patients where the autobahn is just too entrenched, where therapy and medications aren't working, what we call treatment-resistant depression, we have actual physical interventions, right?
Yes, brain stimulation therapies, electroconvulsive therapy, or ECT, and transcranial magnetic stimulation, TMS.
What do those do?
When the brain is profoundly stuck, These modalities use electrical currents or targeted magnetic fields to manually depolarize the neurons en masse.
Just a huge electrical wave.
It is a massive reset that forces the release of BDNF and jumpstarts structural plasticity, essentially forcing the brain to grow out of its stagnant state.
This is all incredibly hopeful for clinical treatment. But I want to pivot to you, the listener, right now.
Good idea.
Not everyone listening has a severe mood disorder, and we certainly don't all have access to or need ketamine infusions or magnetic brain stimulation.
Right.
But the most exciting part of this research is that every single person listening has free, immediate daily access to 3 of the most powerful plasticity modifiers known to human science.
The big 3.
This brings us to the final section, the ultimate brain hacks, how sleep, exercise, and diet physically rewire you.
These are the absolute non-negotiable fundamental pillars of brain health. You can take all the supplements in the world, but if you ignore sleep, physical activity, and what you ingest, you are fighting a losing battle against your own biology.
Let's start with the most critical one, sleep. The literature describes sleep as the permissive state for neural reorganization. Permissive state. That implies that without it, the brain is basically not allowed to change.
That is exactly what it means. We tend to think of sleep as a passive process like powering down a computer to save battery. But neurologically, sleep is an incredibly violent, active period of biological construction and demolition.
Wait, demolition?
Yes. While you are awake, you acquire information. You read an article, you practice a skill, but the actual physical stabilization and integration of those new synaptic connections, the true consolidation of memory, happens predominantly while you are unconscious.
And one of the most fascinating mechanisms driving this involves the expression of a protein called Homer 1A.
Yes, Homer 1A.
I have to ask, did they name a crucial brain protein after a cartoon character?
It sounds like it, doesn't it? But its function is completely serious and critical. Homer 1A proteins act as a molecular integrator of your waking arousal and your sleep state.
What do they do?
During deep sleep, Homer 1A drives a process called synaptic scaling down.
Wait, scaling down? I thought the whole point of sleep was to solidify the connections we built during the day. Why would we scale them down?
Think about it energetically. If your brain only ever scaled up and strengthened every single synapse that got stimulated during your waking hours, your skull would run out of physical space. And your brain would consume more energy than your body could possibly produce. The network would become saturated with meaningless metabolic noise.
Like trying to listen to a symphony where every instrument is playing at maximum volume all the time.
Precisely. So Homer 1A facilitates a homeostatic weakening of synapses across the board while you sleep.
Across the whole brain.
Yes. This global scaling down acts as a massive reset. It proportionately reduces the strength of all connections. The weak, trivial connections from the day fade away entirely.
What about the important stuff?
The strong connections, the things you focused intensely on, survive the scaling down process. It is literally cleaning the slate, removing the background noise so the true signal shines through, and preparing the brain to have the capacity to learn again the next day.
That perfectly explains a phenomenon I think every single person listening has experienced.
What's that?
Have you ever been staring at a problem at work, or a crossword puzzle, or trying to write an email, and your brain is just totally jammed? You cannot see the answer. You finally give up, go to sleep, and when you wake up the next morning, the answer is instantly, blindingly obvious.
Oh, absolutely.
That is not just you being refreshed, that is Homer 1A and sleep-induced plasticity actively at work. It literally swept away the junk connections during the night, so the real pathway was clear.
That is exactly what is happening. Now consider the catastrophic flip side sleep deprivation.
What happens when we don't sleep?
When you restrict sleep, the impact on your neuroplastic potential is devastating. Just 24 hours of sleep deprivation significantly reduces cerebral blood flow.
Wow.
It drastically lowers the production of BDNF, our brain fertilizer. But most dangerously, it creates a severe imbalance in cortical excitability and inhibition.
The sources mention some very specific, highly technical markers for this, CKI and CSP. Can we translate those into what they actually feel like for the listener?
Let's do it. CCI stands for short interval intracortical inhibition, and CSP is the cortical silent period. Essentially, these are measures of the brain's braking system, mostly mediated by the inhibitory neurotransmitter GABA.
Okay.
When you are severely sleep deprived, these inhibitory measures plummet. Your brain's brakes fail.
So the neurons are just firing wildly with nothing to stop them.
Yes. The brain becomes diffusely overexcitable. This is why when you are exhausted, you feel wired and tired.
Oh, that makes so much sense.
Your attention fractures, you are easily startled, your reaction times slow down because there's too much noise in the system. Your brain entirely loses its capacity to induce meaningful, targeted neuroplasticity because it cannot filter anything out.
And what about dreaming? What role does REM sleep play in all this?
REM sleep, the phase where we dream vividly, is heavily involved in the active pruning and maintenance of newly formed synapses, particularly those involving complex, emotional, and associative memories.
Okay, so dreaming is structural work too.
Yes. If you chronically curtail your sleep waking up to an alarm after only 5 or 6 hours, you are disproportionately robbing yourself of REM sleep, which happens mostly in the final hours of the night.
So you missed the most important part.
You are literally destroying your ability to consolidate complex memories.
The takeaway there is brutal, but necessary. Pulling an all-nighter to study for a test or finish a project is biologically counterproductive. You are actively sabotaging the exact mechanism you need to retain the information you are staying awake for.
It's the worst thing you can do for learning.
Let's move to the second pillar, exercise. We all know we should exercise to keep our hearts healthy and our waistlines in check, but the sources frame aerobic exercise as literal structural brain building. How does moving your legs build your brain?
It is a profound catalyst for structural plasticity, and it circles back to our favorite molecule, BDNF. But the research introduces a crucial nuance here that we haven't touched on yet.
What's that?
BDNF doesn't just exist in one form. It exists in 2 opposing hydrolytic forms, pro-BDNF and mature BDNF, or MBDNF.
Opposing forms. So there's a good BDNF and a bad BDNF.
In a simplified way, yes, they generate entirely opposite biological effects. Pro-BDNF binds to a specific receptor called the P75 neurotrophin receptor.
And what happens then?
When it does, it induces long-term depression, promotes amyloid-like degeneration, and actively triggers neuronal apoptosis, which is programmed cell death.
Wait, so pro-BDNF is the grim reaper for brain cells. It kills them.
Yes. Now, pruning and apoptosis are natural, necessary processes in a healthy brain to clear out damaged cells. But in pathological states like chronic stress, severe depression, or chronic pain conditions, the balance shifts dangerously.
Too much of the reaper.
Exactly. The brain starts producing too much pro-BDNF, leading to massive structural atrophy.
What about mature BDNF?
Mature BDNF, on the other hand, binds to the TRKB receptor. This is the protective form. It promotes long-term potentiation, cell survival, and the robust growth of new dendrites and axons.
And I'm guessing exercise tips the scale.
Vigorously. Aerobic exercise strongly shifts the ratio away from the destructive pro-BDNF and heavily toward the protective, growth-promoting MBDNF.
How does it do that?
The mechanism by which it does this is a stunning example of the mind-body connection. It actually involves your liver.
Wait, hold on. My liver? What does my liver have to do with the synapses in my head?
It is an incredible metabolic pathway. When you engage in sustained aerobic exercise, like going for a run or a fast bike ride, your body starts burning fat for energy. As a byproduct of this fat metabolism, your liver produces and releases a ketone body called beta-hydroxybutyrate.
Okay, so the liver pumps out beta-hydroxybutyrate into the blood.
Yes. This molecule travels through your bloodstream, and importantly, it is capable of easily crossing the blood-brain barrier. It enters the brain and heads straight for the hippocampus.
The memory center again.
Once inside the neurons of the hippocampus, beta-hydroxybutyrate acts as a direct inhibitor of histone deacetylase.
Histones, we talked about this. They are the spools the DNA is wrapped around.
Precisely. By inhibiting the enzymes that tighten those spools, the ketone body from your liver forces the chromatin to remain open and relaxed.
What gene is sitting right there ready to be read?
The gene for BDNF.
Wow.
The beta-hydroxybutyrate from your liver directly ramps up the transcription and production of protective BDNF in your brain.
That is absolutely phenomenal. I want everyone listening to visualize this. You go for a jog. Your muscles demand energy. Your liver breaks down fat and sends a chemical text message through your blood directly into your brain, past the cellular walls into the nucleus, and commands your DNA to manufacture more fertilizer to grow memory circuits. That is magic.
It is the biological reality of exercise, and it has profound clinical applications. The sources highlight the impact of this on chronic low back pain.
How does brain plasticity relate to back pain?
Chronic pain is not just a physical sensation happening in your lower back. When a pain signal is constant and unrelenting, it fundamentally alters the brain's macroscopic structure.
Like the stress we talked about earlier.
Very similar. Patients with chronic low back pain often suffer from severe pain catastrophizing, fear of movement, and deficits in executive attention. When researchers scan their brains, they find a literal reduction in cortical thickness in the dorsolateral prefrontal cortex, the DLPFC.
The DLPFC, we mentioned that earlier. That is the area responsible for cognitive control and regulating emotional responses. So the constant pain signal is physically thinning out the brain's armor.
Exactly. The brain is losing its ability to regulate the distress. But clinical studies have demonstrated that when these patients engage in regular aerobic exercise, they don't just feel better physically.
What happens to their brain scans?
The exercise actually physically increases the cortical thickness in their DLPFC.
It rebuilds the armor.
Yes. It reverses the structural atrophy, enhances neural connectivity, and directly combats the cognitive and emotional deficits. Exercise is quite literally restructuring the brain to better manage and quiet the pain signals.
Incredible. So we have sleep clearing the noise and exercise signaling the DNA to build. Let's hit the final pillar: diet and everyday chemicals. What are we putting into our bodies that impacts our neuroplastic potential on a daily basis?
The source material provides some fascinating visual data on the pharmacological impacts of the most widely consumed neuroactive substances on the planet. I am talking about caffeine and its related chemical cousins, paraxanthine, theobromine, and theophylline.
Okay, I am literally holding a cup of coffee right now. Do not tell me it is destroying my neuroplasticity. Tell me exactly what I'm doing to my brain.
You will be happy to hear that in moderation, you are actually priming it.
Oh, thank goodness.
When you drink coffee, you are actively manipulating a specific neurotransmitter system involving adenosine.
Adenosine, what does that do?
Adenosine is an inhibitory neurotransmitter. Throughout the day, as your neurons burn energy, adenosine slowly builds up in the synapses. It binds to specific receptors and slows neural activity down.
So it makes you tired.
It is the chemical that creates sleep pressure, that heavy, tired feeling you get at the end of the day.
So it is the chemical telling me it is time to go to bed.
Exactly. Now, caffeine and its major metabolite paraxanthine are molecularly very similar in shape to adenosine.
Okay, so they look like the key.
Right, so they slip into the synapse and bind to the adenosine A1 receptors, but they don't activate them, they act as antagonists. They block the receptor completely.
Like putting a fake key in a lock so the real key can't get in.
Perfect analogy. By blocking the A1 receptor, caffeine prevents the inhibitory, sleep-inducing signal from registering.
And what happens then?
Because the brakes are artificially blocked, it leads directly to an increased release of excitatory neurotransmitters like glutamate and dopamine throughout the brain. This results in heightened alertness, improved focus, and vasoconstriction.
Okay, so coffee blocks the sleep signal and revs the engine. What about the other compounds? Theobromine is the chemical found in dark chocolate, right?
Yes. Theobromine, along with theophylline, which is found in tea, have a slightly different target. They strongly antagonize the adenosine A2A receptors.
And what does blocking the A2A receptor do?
This is where it gets highly relevant to our topic. Blocking A2A receptors has been experimentally shown to result in enhanced synaptic plasticity, specifically facilitating long-term potentiation.
Are you serious?
Yes. It also improves general cognition and increases locomotion.
So you are telling me that my morning cup of coffee and a piece of dark chocolate are literally chemically priming my synapses to be more plastic and ready to wire new memories?
When used intelligently and in moderation, yes. They create a neurochemical environment that is highly conducive to alertness and synaptic strengthening.
That's amazing.
Of course, if you drink caffeine too late in the day, you disrupt the Homer 1A sleep cycle we just talked about, destroying the consolidation phase. It is all about biological timing.
Which brings this entire deep dive full circle. If we synthesize everything we have discussed today, you have incredible agency over your neural architecture.
You really do.
By combining these three pillars, prioritizing deep, REM-rich sleep so Homer 1A can scale down the metabolic noise, engaging in aerobic exercise so your liver pumps beta-hydroxybutyrate across the blood-brain barrier to unlock your DNA and boost BDNF, and utilizing dietary compounds like caffeine wisely to prime alertness, you are doing something profound.
You are actively creating the absolute optimal biological environment for your brain to heal, to adapt, and to learn.
You are providing the raw building blocks the metabolic energy, and the permissive biological state required for the genomic and structural architectures of your brain to remodel themselves in a positive direction.
You are facilitating your own evolution.
And that is the message I want to bring directly back to you, the listener. If there is one single thing you take away from this conversation, let it be this. You are the architect of your own adaptability. The myth of the static brain is dead and buried.
It's gone.
Every single book you choose to read, every run you push yourself to take, every mindful breath you practice to redirect an anxious, catastrophic thought, and every single night of deep, restorative sleep you get. These are not just fleeting moments.
No, they leave a physical mark.
They are physically, permanently changing the microscopic architecture of your brain. You are continually, relentlessly rebuilding your own mind.
It is a profoundly empowering realization. We are not prisoners of our old neural pathways. We are the engineers capable of directing their reconstruction.
It changes how you view every action you take. But as we wrap up this deep dive, we want to leave you with one final provocative thought derived directly from the furthest edges of the source material.
Oh, this is the really wild stuff.
We have spent this entire time talking about how incredibly plastic our biological brains are, how they constantly rewire themselves to our habits, our traumas, and our physical environments. Now, project that plasticity into the future.
Consider the rapid exploding advancement of Neuralink and brain-computer interfaces, or BCIs.
This is truly where the ancient biology of neuroplasticity collides with the frontier of human evolution. We discussed how adept the brain is at integrating new, bizarre inputs, like the blind individuals who completely rewired their visual cortex to process acoustic click echoes.
If the brain is that hungry to adapt, what happens when we start directly, surgically merging our neural networks with artificial intelligence, silicon chips, and global digital networks?
The sources asked a question that is genuinely mind-bending. When you operate a machine with a BCI, the relationship is entirely mental. There's no mediation of your physical limbs. You just think, and the computer responds.
The researchers suggest this creates a profound psychological and neurological phenomenon of self-disembodiment.
Self-disembodiment. Your mind, your agency, literally extends beyond your biological skull into a digital network.
And it forces us to ask a staggering philosophical question. Will this unprecedented integration stretch our neuroplasticity so far that we fundamentally alter human identity?
If a human mind possesses capabilities, memories, and processing speeds derived from a direct structural neural integration with a machine, do we cease to be strictly human?
Do we transition into a post-human species, a true biological digital cyborg hybrid? And is this radical transformation something to fear? Or is it simply the next logical, inevitable step in the long history of our cognitive evolution?
It is a massive concept to seriously mull over. As our external tools become physically and structurally integrated directly into our internal neuroplasticity, the clear definition of where the human mind ends and the artificial machine begins might just disappear entirely.
It's going to be a fascinating future.
Thank you so much for joining us on this deep dive into the dynamic, shape-shifting, miraculous universe inside your own head. Keep learning, keep consciously rewiring your pathways, and above all, stay curious. We have got a massive, just a staggering stack of research on the table today.
Oh yeah, it's dense.
Very dense.
Yeah.
And it represents this profound paradigm shift in how we understand our own biology. We're pulling from this vast landscape of literature today, everything from the latest updates out of the NIH Brain Initiative to these highly specialized molecular biology papers to clinical psychology reviews.
Reviews that fundamentally redefine mental illness, actually.
Right. And the singular focus of all this disparate data, the reason we are doing this deep dive today, is you, the listener.
Specifically, those 3 pounds of highly metabolic tissue sitting inside your skull.
Exactly. Because for decades, the dominant metaphor in pop science has been to treat the brain like a computer. Like it's this static piece of hardware, a silicon chip where we just upload software in the form of memories or skills or daily habits.
Which is a terrible metaphor, by the way.
It really is.
Yeah.
Looking at the sheer volume of structural imaging and molecular data we have right here, that metaphor isn't just slightly inaccurate, it's completely inverted. The brain is not a static machine at all. It is a living, continuously shifting physical landscape. So the core question we are unpacking today is how our daily experiences,
Mundane stuff, really.
the chronic hum of modern stress, the exact macronutrients we eat, the physical exertion we endure, the chemicals we ingest, how do these inputs not just change our mood, but actually physically rewire the microscopic architecture of our minds.
It's a complete redefinition of human biology. I mean, we are diving into the universe of neuroplasticity today, but we are moving way beyond the usual pop science buzzwords. The literature we're synthesizing frames the brain as an organ entirely built around ruthless adaptation. So our mission today is to explore the granular mechanisms of how the brain alters its physical structure to survive its environment.
And we're going to look at the dark side of that too.
We have to. We'll look at how the brain's attempt to protect itself can lead to severe structural degradation, things that manifest as deep depression and intractable addiction.
Especially regarding dopamine, which we're going to get heavily into.
Oh, the dopamine research is wild. And critically, we'll unpack the emerging science of how you can actively intervene in this process, how you can leverage pharmacology, neurostimulation, and specific daily protocols to hack your own neuroplasticity and build a more resilient mind.
Okay, let's unpack this, because the idea that learning a new skill actually changes the physical weight and shape of the brain is just mind-blowing to me. How does this actually happen at the micro level?
Well, the literature frames it around two main forces: long-term potentiation and long-term depression, LTP and LTD.
And these aren't just simple on and off switches for memory, right?
Not at all. They are incredibly dynamic, competitive forces constantly sculpting our neural networks. If we look at the synaptic level...
Microscopic gaps between the neurons.
Right, those junctions. LTP is the mechanism of encoding. When a presynaptic neuron repeatedly and effectively fires and stimulates a postsynaptic neuron, that connection physically strengthens.
So an receiving neuron basically bulks up.
Exactly. It increases its receptor density and becomes physically more sensitive to future signals. This is the absolute cellular foundation of learning.
But the research really highlights LTD, long-term depression. And I was struck by this because it's usually casually dismissed as just, the brain forgetting things.
Yeah, which is a massive oversimplification.
Right. The molecular data shows it's actually this aggressive, vital survival mechanism. It prevents the network from just getting totally saturated.
That is a crucial distinction, because if a neural network only had LTP, if it only strengthened, the system would face catastrophic failure.
It would just overload.
Runaway excitation, metabolic exhaustion, and eventually excitotoxicity.
Excitotoxicity.
Yeah, where the neurons literally fire themselves to death. So long-term depression is this activity-dependent reduction in synaptic efficacy. It actively prunes irrelevant connections to maintain synaptic homeostasis.
It's like a city grid. Traffic, or neural activity, dictates which roads get widened into massive multi-lane highways.
That's the LTP.
Yeah. And the neglected dirt roads just get abandoned and paved over. That's the LTD.
Yeah.
It ensures the network stays efficient.
Exactly. It prioritizes the pathways that matter for survival and degrades the ones that are merely noise.
I found the mechanics of how the cell actually initiates this process fascinating. Specifically regarding the NMDA receptor. The papers describe it essentially as a molecular AND gate, a coincidence detector.
It's a marvel of evolutionary engineering, honestly. It's a glutamate receptor sitting on the postsynaptic membrane. But unlike standard receptors that just open up the second their neurotransmitter binds to them, the NMDA receptor is gated by a voltage-dependent physical block.
A physical plug.
Yes. At the neuron's resting membrane potential, the pore of the NMDA receptor is plugged incredibly tightly by a single magnesium ion.
So even if glutamate, which is the chemical key, binds to it perfectly,
the channel remains completely blocked. It requires the postsynaptic membrane to undergo a significant electrical depolarization.
And where does that electrical charge come from?
Usually from neighboring AMPA receptors getting activated at the exact same time. Once the internal voltage of the cell rises enough, the positive charge forcefully repels that positively charged magnesium ion. It literally ejects it from the pore.
So it requires both the chemical key of glutamate A and D, a strong electrical surge to blow the magnesium plug out.
That's the coincidence it's detecting. And once that happens, the data shows a massive influx of calcium ions flooding into the cell.
Which acts as the ultimate biological trigger for construction, right?
The pivotal moment, yeah. Calcium is a powerful secondary messenger. It binds to something called calmodulin, which then activates an enzyme called CAMKII.
The molecular construction form.
Perfect way to describe it. It triggers this massive intracellular cascade that leads to the rapid insertion of more AMPA receptors into the membrane, and even changes gene expression to physically enlarge the dendritic spine.
That single calcium flood is the brain's physical way of deciding that a specific connection is vital and have to be reinforced.
Exactly, and that structural reinforcement maps perfectly onto Hebbian plasticity.
The old axiom, neurons that fire together wire together.
Right. But the literature highlights a massive vulnerability in Hebbian theory. If you rely solely on firing and wiring, you create an endless positive feedback loop.
Because the connection gets stronger, which makes it easier to fire.
Makes it even stronger.
Leading to that runaway excitation you mentioned, the excitotoxicity.
Right. And this is where the concept of metaplasticity completely changes the picture.
Metaplasticity, the plasticity of plasticity.
It's one of the most elegant regulatory systems in our biology. The brain has these built-in homeostatic mechanisms that continuously adjust the threshold required to induce either LTP or LTD.
Based on what?
Based on the prior history of that specific neuron's activity. If a neuron has been highly active, metaplasticity raises the threshold for future LTP. It makes it harder to strengthen connections further, while lowering the threshold for LTD, making pruning easier.
Wow, and if it's been quiet?
The system makes LTP easier to achieve. It's a sliding scale. It ensures the neural network always operates within a safe, functional, dynamic range to prevent structural instability.
Now, the raw material driving all this physical growth, the actual fertilizer, is brain-derived neurotrophic factor, BDNF.
Oh, BDNF is critical.
It's heavily featured in the research. But there's a profound nuance here that seems entirely glossed over in normal science communication. The data clearly shows BDNF is not a universal force for growth.
No, not at all.
It exists in two completely antagonistic forms.
The dichotomy of BDNF is a perfect example of how complex these systems are. The molecule is initially synthesized as a precursor protein called pro-BDNF.
And this is the vital nuance, right?
Yes, because pro-BDNF and its mature form, MBDNF, elicit entirely opposing biological responses. When pro-BDNF is secreted, it binds to a receptor called P75.
And what does that do?
Activation of the P75 receptor actively promotes long-term depression. It triggers the retraction of dendritic spines. It can even induce neuronal apoptosis.
Programmed cell death. So the precursor is actively dismantling the network.
It acts as a targeted demolition signal. However, pro-BDNF can be cleaved by extracellular proteases, enzymes that basically snip the protein,
converting it into mature BDNF.
MBDNF.
Exactly. And this mature form has a high affinity for a completely different receptor, the TRKB receptor. Binding to TRKB initiates the neuroprotective effects.
It promotes the robust LTP, stimulates neurogenesis, and grows new dendrites.
Right. So the adaptability and health of your brain aren't merely determined by how much total BDNF you produce. It's governed by the highly regulated microscopic balance between the demolition signal of pro-BDNF and the growth signal of MBDNF.
That microscopic dance is happening millions of times a second, constantly optimizing the network based on the environment we navigate. But that brings us to a rather dark realization in the literature.
The maladaptive side.
Yeah, because if the brain is this hyper-responsive to environmental inputs, what happens when the environment is objectively hostile, inescapable, chronic?
The data on maladaptive plasticity is deeply unsettling. We often frame neuroplasticity as this inherently positive, self-optimizing feature. But the brain is completely value neutral, isn't it?
Totally value neutral. It simply adapts to survive. Even if that adaptation destroys our cognitive baseline, the clinical data on chronic stress provides the clearest window into this.
Let's talk about the HPA axis.
The hypothalamic pituitary adrenal axis. When you perceive a threat, the hypothalamus releases corticotropin-releasing hormone. That signals the pituitary gland, which signals the adrenal glands to flood your system with glucocorticoids, cortisol.
Which is highly adaptive in the short term. It mobilizes glucose, focuses your attention so you don't get eaten by a tiger.
But the system evolved to handle acute, transient threats, the architecture of modern life.
The incessant digital communication, the constant financial precarity, the terrible sleep.
All of it forces the HPA axis into a state of chronic, unresolved activation.
And the prolonged exposure to those glucocorticoids stops being a performance enhancer and becomes severely neurotoxic.
Highly toxic.
I was looking at the structural imaging data regarding the hippocampus, and it is genuinely haunting. We usually treat major depression or chronic anxiety as purely psychological phenomena, or just invisible chemical imbalances.
But the MRI data shows undeniable structural degradation. The hippocampus is exceptionally vulnerable to glucocorticoid toxicity because it is packed with receptors for these specific hormones.
So under a chronic chemical flood, what actually happens to it?
It undergoes profound morphological changes. We observe severe dendritic atrophy, particularly in the apical dendrites of the CA3 pyramidal neurons.
The branches literally shrink.
They retract and shorten by 20 to 30%. And furthermore, the chronic stress completely halts neurogenesis, the birth of new neurons, in the dentate gyrus.
A 20 to 30% retraction of the receiving branches. The physical volume of the hippocampus shrinks. The studies report up to a 24% volume loss in patients with borderline personality disorder.
And similar, massive reductions in patients with severe treatment-resistant depression.
It perfectly contextualizes the clinical symptoms. The pervasive brain fog, the inability to form new episodic memories, the feeling of being cognitively anchored to a single dark state.
Because the physical hardware required to process new contexts and learn new emotional responses has literally withered away.
But the tragedy of this maladaptive plasticity is compounded when you look at what's happening to the amygdala at the exact same time.
Right, the contrast is stark. While the hippocampus is undergoing severe atrophy, the amygdala, the brain's primary threat detection and fear processing center, is experiencing the exact opposite phenomenon.
Drawing.
Under the same chronic glucocorticoid exposure, the neurons in the basolateral complex of the amygdala undergo hyperplasticity. They increase their dendritic arborization, they form new, highly excitable synapses.
So the brain is actively upgrading its threat detection hardware while simultaneously dismantling its logic and memory centers.
It's a devastating structural realignment. The result is a neural architecture that is incredibly efficient at detecting danger, wildly hypervigilant, but completely lacks the hippocampal volume required to contextualize that danger.
It can't realize that a threat has actually passed. It just stays panicked.
Exactly. And the timeline for this kind of physiological rewiring can start alarmingly early. The epidemiological data from the World Health Organization regarding early life stress is staggering.
Yeah, they define early life stress as sustained adverse experiences, profound neglect, extreme poverty, exposure to violence, occurring anywhere from birth to 18 years of age.
And they estimate that 1 billion adolescents are exposed to these conditions globally every year. 1 billion.
The literature shows that exposing a developing brain to that level of sustained HPA axis activation fundamentally alters its developmental trajectory. The regulatory feedback loops become compromised.
They basically get locked in the on position. We see epigenetic changes, specifically DNA methylation, that permanently alter how genes related to stress resilience are expressed.
And structurally, we see decreased gray matter volume and a severely compromised prefrontal cortex.
And the downstream behavioral consequences of that early structural deficit are massive, particularly regarding the propensity for addiction.
Which is a huge theme in this stack.
When the brain's internal regulatory systems are structurally degraded early in life, the organism naturally seeks external, highly potent chemical regulation to stabilize its internal state.
The data indicates that experiencing multiple adverse childhood events exponentially increases the statistical probability of severe substance use disorders and alcohol dependence in adulthood.
And when we look at the cellular environment driving these depressive and addictive behaviors, the research highlights a massive paradigm shift. For decades, neuroscience was almost entirely neurocentric.
Just focused on the neuron.
Right, but the literature here demonstrates that glial cells, specifically astrocytes and microglia, are the hidden architects of this maladaptive state.
This reevaluation of glial cells is incredible. Astrocytes are supposed to provide vital metabolic and structural support to neurons.
But during chronic stress, they actively undergo atrophy in the prefrontal cortex. They retract their processes, and they fail to clear glutamate from the synaptic cleft.
Which contributes to that excitotoxicity we discussed earlier.
Exactly. And simultaneously, microglia, which are the resident immune cells of the central nervous system, shift from a resting, surveying state into a highly aggressive, activated phenotype.
This leads right into the cytokine hypothesis of depression. The data shows these activated microglia just pouring out pro-inflammatory cytokines, specifically tumor necrosis factor alpha, or TNF-alpha.
Creating a localized state of intense neuroinflammation. This inflammation actively suppresses neurogenesis and drives behavioral despair.
It reframes severe depression. It's not just a structural issue or a chemical imbalance. It's essentially an autoimmune-like inflammatory disease of the brain.
It really is. And building on that inflammatory model, we can bridge directly into how addictive substances and really any unnatural dopamine spikes weaponize these exact mechanisms.
Let's dive deep into this. The hijacked reward system. Why does our brain chase dopamine so strongly? Because the dopamine research here, specifically regarding modern digital addiction and substance abuse, is mind-blowing.
Well, to answer why we chase it, you have to look at evolution. The literature on the mesolimbic pathway, specifically the dopaminergic projections from the ventral tegmental area to the nucleus accumbens.
The VTA to the NROC.
Right. This circuitry highlights an evolutionary mismatch. Our brains evolved this intricate reward system to reinforce rare, highly effortful behaviors that were absolutely critical for survival.
Finding calorie-dense food in a famine, navigating to safe shelter reproduction.
Exactly. Dopamine acts as the ultimate teaching signal for survival.
But the research emphasizes that dopamine isn't just the pleasure chemical that pop culture makes it out to be, right?
Not at all. The classic experiments involving primate learning show that dopamine neurons spike not when a reward is received, but in anticipation of a predicted reward.
It is a molecule of craving, of motivation.
And most importantly, a structural reinforcement. It instructs the NMDA receptors to aggressively hardwire the specific sequence of actions that led to the reward.
There's literally building the physical habit loops.
Yeah.
And that is exactly where the evolutionary mismatch becomes lethal in the modern world.
Lethal is the right word.
Because modern addictive drugs, or even hyper-engineered digital stimuli,
Like infinite social media feeds, TikTok, Instagram.
right, they hijack this exact system. They deliver a highly concentrated, completely unnatural surge of dopamine that completely overwhelms the brain's homeostatic limits.
A massive dopamine flood, and this flood forcefully induces widespread aberrant synaptic adaptations in the nucleus accumbens.
It's rogue plasticity.
Addiction at the cellular level is a form of rogue, drug-evoked neuroplasticity. The brain's survival architecture is basically tricked into prioritizing the acquisition of the drug or the digital hit above all other biological imperatives.
And there's this wildly counterintuitive study in the stack regarding this process. It looks at cocaine sensitization and microglial activation. You would assume the brain's immune system just suffers under the chemical assault.
But the data shows it actually mounts a targeted defense.
A physical defense against the addiction.
Yes. The immune response to cocaine is a fascinating physiological countermeasure. Researchers discovered that repeated cocaine administration robustly activates the striatal microglia.
The same immune cells driving inflammation and depression.
The very same. And just as we saw in the depression models, these activated microglia synthesize and release the inflammatory cytokine TNF-alpha. But in the context of the nucleus accumbens and cocaine,
This localized burst of TNF-alpha actually initiates a protective form of long-term depression.
it actively depresses and weakens the glutamatergic synaptic strength. It is effectively trying to limit the development of behavioral sensitization to the drug.
The brain's immune system recognizes the runaway rogue plasticity as a pathogen. It actively releases inflammatory agents to degrade the structural highways the cocaine is trying to build. That is incredible.
And the twist in the study regarding abstinence completely reframes our understanding of relapse and craving.
What happens when they stop?
The researchers found that after a period of prolonged abstinence, the microglia return to a morphologically resting state, but they remain biochemically primed.
Like they remember the addiction.
Exactly. And if the subject is then exposed to a very mild, non-specific immune trigger,
They used a TLR4 agonist in the study, right? Which just simulates a mild bacterial infection.
right. When exposed to that mild immune trigger, these primed microglia rapidly reactivate. They dump TNF-alpha back into the striatum, depress the synaptic strength again, and successfully suppress cocaine-seeking behaviors long after the drug use has stopped.
So a mild peripheral immune response could theoretically suppress neurological craving because the microglia possess an immunological memory of the addiction.
It's a massive leap forward in how we view control and reset mechanisms in the brain.
And speaking of massive leaps, the data on the lateral habenula fundamentally alters how we view the physical roots of anhedonia. If the VTA is the engine of motivation and dopamine,
The lateral habenula is the ultimate emergency brake.
it's this small epithalamic structure that has become central to the modern understanding of major depressive disorder, the brain's anti-reward center.
When an organism anticipates a reward and the reward is withheld, or when the organism experiences a highly aversive, punishing stimulus, the lateral habenula fires intensely.
And its projections travel down to the VTA and the raphe nuclei, actively suppressing the release of both dopamine and serotonin.
The structural data here is profound. In clinical models of severe depression, the lateral habenula exhibits massive hyperactive plasticity,
The excitatory synapses are strengthened.
and the region becomes chronically overactive. It is continuously firing, keeping the brakes permanently clamped down on the brain's motivational and mood-regulating centers.
So the inability to feel pleasure, that crushing weight of anhedonia, isn't just a passive absence of serotonin or dopamine.
It is an active, aggressive suppression driven by a hyperplastic anti-reward center.
And the specific cellular mechanism driving this hyperactivity brings us right back to the glia.
It does. The literature highlights the role of astroglial KIR 4.1 channels in the lateral habenula.
These inward rectifying potassium channels expressed exclusively on the astrocytes surrounding the neurons.
In depressive states, these channels become severely upregulated. This massive increase in potassium buffering completely destabilizes the extracellular environment.
It forces the nearby neurons into an abnormal, rapid burst-firing pattern.
The astrocytes are effectively short-circuiting the neurons, driving the very hyper-excitability that shuts down the reward system.
Okay, so let's summarize where we are. We are looking at a system that is physically degraded. We have a shrunken hippocampus, an overgrown, hypervigilant amygdala, inflamed microglia, a hijacked dopamine system driving rogue plasticity, and an anti-reward center locked in a state of rapid-fire suppression by its own support cells.
It paints a grim picture.
It really does. So the critical question transitioning into the clinical data is, how do we force this deeply entrenched hostile architecture to dismantle itself and rebuild? How do we rewire for recovery?
Well, the traditional pharmacological approach has always relied on the slow, agonizing process of gradual neurogenesis. Standard SSRIs like fluoxetine or Prozac.
Why do they take weeks or months to work?
Because they operate by inhibiting the reuptake of serotonin, which instantly floods the synapse with the neurotransmitter. But the clinical efficacy lags because the serotonin itself is not the cure.
It's merely the first slow signal.
Right. It's the start of a massive downstream cascade that eventually leads to an upregulation of mature BDNF and the slow, arduous birth of new neurons in the dentate gyrus.
It's a grueling timeline when a patient is in acute crisis.
Exactly. But the literature on psychoplastogens, these compounds capable of inducing rapid structural neuroplasticity, represents a true paradigm shift.
The data on subanesthetic infusions of ketamine, it is nothing short of revolutionary. We are talking about reversing severe structural atrophy not in months, but in hours.
The mechanism of action for ketamine is a masterclass in exploiting the brain's complex circuitry. Ketamine is an NMDA receptor antagonist.
It blocks the very receptor we said was crucial for learning.
But at low clinical doses, it exhibits a profound preference. It selectively blocks the NMDA receptors located specifically on inhibitory GABAergic interneurons in the prefrontal cortex.
And these interneurons normally act as localized brakes, constantly suppressing the activity of the surrounding excitatory pyramidal neurons.
So by selectively blocking the inhibitor, ketamine essentially cuts the brake lines.
The prefrontal cortex experiences a massive, immediate surge of excitatory glutamate.
Precisely. This sudden, robust glutamate surge bypasses the blocked NMDA receptors and violently activates AMPA receptors.
And what does that intense AMPA activation do?
It triggers the release of mature BDNF and initiates a profound intracellular signaling cascade involving the mammalian target of rapamycin, the mTOR pathway.
Activation of mTOR is the biological equivalent of throwing a massive switch for cellular construction.
It immediately begins the synthesis of new synaptic proteins. Researchers utilizing advanced two-photon microscopy have observed the physical growth of new functional dendritic spines in the prefrontal cortex within mere hours of administration.
It rapidly rebuilds the exact synaptic connections that chronic stress and toxic cortisol levels destroyed. The idea of medically induced hyperaccelerated structural remodeling is incredible.
It is.
But the literature pushes this even further when discussing the concept of critical periods. Here's where it gets really interesting. We all recognize that early childhood is a time of terrifyingly efficient neuroplasticity.
Kids effortlessly wire complex motor patterns. They acquire entirely new phonemic structures for language like it's nothing.
But as adults, that level of fluid adaptability seems entirely lost.
Because the brain actively restricts massive structural plasticity as we age. It has to preserve essential functional stability.
Once the foundational circuits of the visual, auditory, and motor systems are established, the brain deploys these potent molecular brakes.
It downregulates certain growth factors and literally wraps mature neurons in dense extracellular matrix structures called paraneuronal nets.
Paraneuronal nets. They physically lock the synapses in place, severely limiting further structural changes. The critical period closes and the architecture solidifies.
But the data suggests we can artificially dissolve that solid architecture.
The visual cortex study involving fluoxetine.
Right. Researchers were able to reopen ocular dominance plasticity in fully mature adult subjects. Their visual systems reverted to a state of hyperadaptability comparable to a developing child.
The fluoxetine study proved that the breaks are not permanent.
Yeah.
They are actively maintained and can be pharmacologically disrupted.
However, that specific intervention was highly transient. The plasticity window remained open only as long as the drug and its active metabolites were present in the cerebrospinal fluid.
Once cleared, typically within 7 days, the paraneuronal nets restabilized and the critical period slammed shut again.
Which sets the stage for the most astonishing proof of concept trial in this entire stack.
The Valproate Absolute Pitch Study. Absolute pitch, the ability to identify or recreate a musical note without a reference tone, is the textbook example of a trait bound by a strict critical period.
The consensus has always been that if you don't receive targeted musical training before the age of 7, the auditory cortex loses the capacity to wire that specific skill.
It was considered a biological impossibility for an adult.
Until the application of Valproate. Valproate is traditionally utilized as an anticonvulsant and mood stabilizer, but fundamentally it operates as a potent epigenetic modulator.
Specifically, it acts as a histone deacetylase inhibitor.
Inside the cell nucleus, DNA is tightly wound around structural proteins called histones. This tight spooling prevents the cellular machinery from accessing and reading specific genes.
By inhibiting histone decetylation, Valproate forces the chromatin to uncoil.
It exposes vast arrays of genes, specifically those related to profound structural neuroplasticity that had been silenced since childhood.
And the methodology of this study was incredibly precise. They recruited adult men with absolutely no absolute pitch ability.
They titrated the Valproate to maintain a highly specific serum concentration between 350 and 700 micromoles per liter, ensuring the epigenetic modulation was active without inducing toxicity.
And while maintaining that specific blood concentration, they subjected the men to an intensive 2-week absolute pitch training protocol.
The results shattered previous neurological assumptions. The cohort receiving the Valproate performed significantly better than the placebo group. They successfully acquired foundational aspects of absolute pitch.
The drug systematically degraded the molecular brakes, transiently returning the adult auditory cortex to a juvenile, highly plastic state. They chemically rolled back the developmental clock.
To allow a mature brain to absorb a skill that was supposedly lost to time, it's a breathtaking proof of concept.
It really is. But the literature isn't solely focused on pharmacology. The advancements in non-invasive, targeted physical interventions are producing remission rates that rival the most potent chemical treatments.
The data on transcranial magnetic stimulation, particularly the Stanford SAINT protocol, it completely redefines psychiatric intervention.
TMS operates on the principle of electromagnetic induction. By placing a specialized magnetic coil over the scalp, clinicians can deliver highly focused magnetic pulses that penetrate the skull.
And induce highly specific, localized electrical currents in targeted cortical regions. By precisely modulating the frequency and pattern of these pulses, they can force a specific brain network to undergo either LTP or LTD.
And what the team at Stanford did with the same protocol was hyper-optimize this process. They didn't just guess where to aim the magnet.
No, they utilized accelerated functional MRI to map the unique neural topography of each individual patient.
They specifically located the exact coordinates in the dorsolateral prefrontal cortex that exhibited the strongest negative functional connectivity to the subgenual anterior cingulate cortex.
A region deeply implicated in the pathology of severe depression. It is the absolute pinnacle of personalized precision neuroengineering.
Once they mapped that exact target, they delivered a highly accelerated, incredibly dense schedule of intermittent theta burst stimulation.
They compressed months' worth of standard TMS treatment into just 5 intensive days.
And the clinical outcomes were unprecedented. In a cohort of patients suffering from severe treatment-resistant depression, individuals who had failed multiple pharmacological trials,
nearly 80% achieved full clinical remission within days.
They structurally rebuilt the regulatory networks of the brain using nothing but focused magnetic fields.
And we are seeing the same ethos of precision engineering applied to neurodegenerative disorders. The NIH Brain Initiative data highlights the leap from traditional deep brain stimulation to adaptive deep brain stimulation, or ADBS, for Parkinson's disease.
Traditional DBS is kind of a blunt instrument. It involves surgically implanting electrodes into specific basal ganglia targets and delivering a constant, unvarying electrical pulse.
To disrupt the pathological firing patterns that cause tremors and rigidity.
But adaptive DBS incorporates advanced closed-loop technology.
The implanted system continuously records the patient's localized field potentials, the real-time electrical chatter of the brain.
It utilizes highly sophisticated algorithms to instantly analyze those signals and dynamically adjust the amplitude and frequency of its stimulation.
Millisecond by millisecond, delivering exactly the amount of current required precisely when the brain needs it.
It is a fully autonomous, intelligent pacemaker for the brain, seamlessly integrating with the patient's shifting neurophysiology. The trial data shows it significantly outperformed traditional DBS, reducing the most distressing motor symptoms by an additional 50%, while dramatically lowering the energy output and side effects.
It's incredible.
But we have to bring this incredible high-level science back down to the reality of listener. How do we hack our own plasticity?
Right, because we cannot all access fMRI-guided magnetic helmets, closed-loop brain implants, or sub-anesthetic ketamine infusions this week.
The critical transition here is looking at the literature regarding endogenous plasticity. The data clearly demonstrates that our daily mundane behaviors exert a massive structural influence on our neural architecture.
If we look at the research on physical exercise, it's essentially described as an endogenous psychoplastogen.
The ultimate hack. The clinical framing of exercise must shift from pure cardiovascular or metabolic maintenance to active structural brain remodeling.
When an individual engages in acute high-intensity aerobic exercise, they initiate a systemic, multi-organ chemical cascade that directly targets the central nervous system.
The data reveals that a single session of vigorous aerobic output can induce a 32 to 45% surge in circulating levels of mature BDNF.
A 45% increase in the brain's primary growth factor, generated entirely by physiological exertion.
And the mechanism connecting the peripheral body to the brain during exercise is deeply fascinating, specifically the liver-brain axis.
The liver plays a surprisingly critical role in neuroplasticity. During intense, sustained physical exertion, hepatic glycogen stores are depleted, and the liver shifts its metabolic profile to begin oxidizing fatty acids.
This process results in the robust production of ketone bodies, specifically beta-hydroxybutyrate.
This molecule is highly soluble, rapidly enters the systemic circulation, and easily crosses the blood-brain barrier.
And once it enters the brain, particularly the hippocampus, it doesn't just act as an alternative fuel source. The data shows it acts as a highly potent epigenetic regulator.
Exactly. Beta-hydroxybutyrate functions as an endogenous inhibitor of histone deacetylases, very similar in mechanism to the valproate we discussed earlier.
So by inhibiting these enzymes in the hippocampus, beta-hydroxybutyrate forces the chromatin surrounding the BDNF gene to unspool and remain in an open, transcriptionally active state.
The physical act of running forces the liver to send a direct molecular signal to the hippocampus, commanding it to unlock its DNA and significantly upregulate the production of BDNF,
thereby accelerating synaptic plasticity and neurogenesis. And the profound structural impact of exercise scales up from the microscopic epigenetic level to the macroscopic network level.
The neuroimaging data shows that sustained aerobic exercise fundamentally alters the functional connectivity between the brain's large-scale intrinsic networks, specifically the default mode network and the central executive network.
The default mode network, the DMN, is primarily active during internally directed cognition, daydreaming, autobiographical memory retrieval, and crucially, rumination.
In depressive and highly anxious states, the DMN becomes hyperconnected and dominant. The individual becomes trapped in an endless loop of negative self-referential thought.
The central executive network, the CEN, conversely governs externally directed attention, complex problem solving, and cognitive control.
The literature demonstrates that regular exercise strengthens the white matter tracks, the physical infrastructure connecting these networks.
It enhances the brain's structural capacity to willfully disengage from the ruminative trap of the default mode network and fluidly transition into the focused, engaged state of the central executive network.
You are physically building the roads required to steer your own attention.
And if BDNF is the cellular construction crew building these roads, the nutritional data dictates the quality of the raw materials they are using.
The literature on the gut-brain axis and specific macronutrients reveals that you cannot sustain high-level neuroplasticity on a structurally deficient diet.
The lipid composition of the brain is incredibly specific and relies heavily on dietary inputs. Omega-3 polyunsaturated fatty acids, specifically docosahexaenoic acid, or DHA, are absolutely foundational.
Because the neuronal cell membrane is not a static wall. It is a highly fluid, dynamic lipid bilayer.
The receptors we spent so much time discussing, the NMDA and AMPA receptors, must be able to move, cluster, and embed themselves within this membrane to execute long-term potentiation.
DHA provides the essential biochemical fluidity required for this movement. A diet deficient in DHA results in rigid, inflexible neuronal membranes, fundamentally impairing the physical mechanics of synaptic plasticity.
The data on polyphenols is equally critical, particularly compounds like curcumin found in turmeric. The literature shows that curcumin doesn't just offer mild antioxidant support.
It actively penetrates the blood-brain barrier and engages specific molecular pathways. Curcumin has been shown to robustly activate the AKT-CRB signaling pathway.
CRB is a highly critical transcription factor that, once activated, binds directly to the DNA to massively upregulate the transcription of BDNF.
Furthermore, curcumin exerts profound anti-inflammatory effects within the central nervous system, directly antagonizing the microglial activation and TNF-alpha production we previously identified as the primary drivers of depressive pathology.
And the gastrointestinal tract itself is actively modulating the brain's chemical environment. The gut microbiome is responsible for fermenting dietary fiber into short-chain fatty acids, like butyrate.
These SCFAs interact directly with the enteric nervous system and transmit potent regulatory signals straight up the vagus nerve to the brain, suppressing neuroinflammation and modulating the synthesis of key neurotransmitters.
Which brings us to the most universally consumed psychoactive compound globally, caffeine.
Ah, yes.
The data on how caffeine and its primary metabolites theobromine, paraxanthine, and theophylline interact with the brain's plasticity mechanisms is remarkably nuanced.
Caffeine operates fundamentally as an antagonist to adenosine receptors. As neurons fire and consume ATP for energy throughout the waking day, adenosine accumulates in the extracellular space.
It binds to its receptors, slowly hyperpolarizing the neurons and exerting a profound sleep pressure.
Caffeine's primary mechanism of promoting wakefulness is by competitively blocking the adenosine A1 receptors, which instantly removes this inhibitory brake and triggers a widespread release of excitatory neurotransmitters.
But the literature highlights that its role in structural plasticity is driven by its interaction with an entirely different receptor subtype.
The crucial mechanism for our discussion is its blockade of the adenosine A2A receptors. These receptors are heavily concentrated in the striatum and the hippocampus.
The data indicates that antagonizing the A2A receptors physically lowers the threshold required to induce long-term potentiation.
By blocking this specific receptor, caffeine actively sensitizes the synapses, enhancing structural adaptability and significantly boosting spatial memory consolidation and overall cognitive flexibility.
It is actively lowering the molecular barrier to entry for network construction. But the ultimate prerequisite for all this construction,
the absolute non-negotiable requirement for neuroplasticity,
is sleep. We can flood the brain with BDNF, optimize our DHA levels, and drink all the coffee we want, but the physical reality is that the actual structural remodeling does not occur while we are awake.
Wakefulness is the period of acquisition. We encounter stimuli, activate the NMDA receptors, and initiate the calcium cascades.
But the waking brain is metabolically constrained. It cannot simultaneously process complex environmental inputs and undergo massive structural reorganization.
Sleep is the state of active, aggressive consolidation. During the deepest stages of non-REM sleep, the brain utilizes highly synchronized electrical oscillations.
Specifically, sleep spindles and sharp wave ripples originating in the hippocampus.
It uses those to systematically replay the exact neural firing patterns that occurred during the day.
It replays the day's code, but at a vastly accelerated rate, forcefully cementing the long-term potentiation pathways that represent vital new information.
While simultaneously utilizing long-term depression to widely scale down and prune the synapses that simply represent background noise.
It is the ultimate expression of synaptic homeostasis. If an individual suffers from chronic sleep deprivation, they force their brain into a state of aberrant, chaotic cortical excitability.
The synapses remain saturated from the previous day's inputs. Without the localized pruning and structural cementing that occurs exclusively during deep sleep, the brain is physically incapable of inducing new functional LTP the following morning.
The capacity for learning, emotional regulation, and psychological resilience is structurally compromised.
Totally compromised.
We have mapped an immense and intricate landscape today. We started at the fundamental, microscopic architecture of change, examining the precise voltage requirements of the NMDA receptor and the delicate high-stakes balance between the demolition signals of pro-BDNF and the vital growth signals of mature BDNF.
We navigated the devastating structural reality of maladaptive plasticity. We saw how the chronic, toxic flood of cortisol physically retracts the dendritic branches of the hippocampus, driving memory loss and cognitive rigidity,
while simultaneously forcing the amygdala into a state of hypervigilant panic. We examine how profound early life stress epigenetically degrades the brain's regulatory systems, setting a dangerous trajectory for future addiction.
We explored the intricate mechanisms of the mesolimbic pathway, observing how the evolutionary imperative of dopamine is hijacked by supernormal digital and chemical stimuli, inducing runaway, drug-evoked structural changes.
We witnessed the brain's immune system desperately deploying localized inflammation to fight off this rogue rewiring, and we uncovered the cellular pathology of despair, how hyperactive astrocytes in the lateral habenula actively short-circuit the brain's motivational core.
We also documented the breathtaking frontiers of neurological intervention. We analyzed the rapid mTOR-driven structural regrowth triggered by psychoplastogens like ketamine.
We saw the precision engineering of fMRI-guided magnetic stimulation pulling patients out of intractable depression in days, and adaptive closed-loop implants seamlessly pacemaking the compromised circuitry of Parkinson's patients.
We explored the astonishing reality that epigenetic modulators like valproate possess the sheer molecular force to dissolve paraneuronal nets and roll back the developmental clock, briefly returning the rigid adult mind to the hyperplastic state of infancy.
And finally, we grounded this vast scientific revolution in the undeniable structural power of endogenous plasticity. The realization that an intense aerobic effort forces the liver to chemically command the brain to unlock its DNA and synthesize growth factors.
The knowledge that the structural integrity of our memories relies entirely on the precise lipids we consume and the sanctity of our deep sleep architecture.
The ultimate synthesis of this literature is a mandate of absolute agency. Every specific environment you immerse yourself in, every agonizing physiological effort you endure, every specialized macronutrient you ingest, and every hour of deep consolidation you protect is an active physical instruction to your biological hardware.
You are not a passive observer of a static machine. You are the active architect of a continuously evolving neural landscape. Understanding your brain, your dopamine, your stress, it's the first step to taking control of your attention and your biology.
It is the most empowering realization in modern biology.
It really is. And as we conclude this deep dive, I want to linger on the profound implications of that valproate proof of concept trial. We possess clinical evidence that it is biochemically possible to temporarily dissolve the structural breaks of adulthood and reopen the critical learning windows of childhood, allowing mature individuals to rapidly acquire a neurological trait previously deemed biologically impossible.
It fundamentally destabilizes our concept of fixed adult identity.
It raises an incredibly provocative question regarding the future of human optimization. If the rapid progression of this neuroscience eventually perfects the ability to selectively and safely unlock these highly plastic critical periods on demand, what foundational human traits, what impossibly complex foreign languages, or what entirely novel, high-dimensional ways of perceiving reality might you choose to learn from absolute scratch? If the architecture of the adult mind becomes completely, entirely malleable by